This tutorial is about adding amino acids to a protein. Manually adding amino acids gives you the flexibility to build missing portions of the protein, but be cautious as these added amino acids may not be in the ideal conformation. After adding amino acids, it's essential to perform an energy minimization to return the protein structure to its lowest energy state. To start, load your desired protein model in YASARA as a .pdb file. In this example, we will load a PDB file that is always present in YASARA called 1crn.pdb. If desired, rotate the model to a conformation or a viewing area that pleases you. You may move your cursor down to the bottom of the screen to see the primary amino acid sequence. You can click on this blue dot over on the left to keep this sequence from disappearing as you're working. Upon opening your protein model, it's a good idea to scroll to the far left and the far right of this primary sequence to determine the N and C terminus of the protein. In this case, our N terminus is a threonine. We can use the slider bar to move to the C terminus, which is number 46. All right, let's add an amino acid. First thing to do is to click on the object name here in the heads-up display at the far right. Our object name is 1crn. We'll click here to select it. Right-click on this object name and choose the option Add Residue to Protein. A pop-up window is going to allow you to select the residue you would like to add. You can use the slider bar to see them all. In this example, we're going to add a cysteine, and we're going to add it to the C terminal, so click that button. We will leave the default parameter of the L amino acid configuration. You can choose the angles that you would like the protein to be added, and we will choose Add with above dihedral angles. A cysteine would have been added to the end of our chain. Let's take our slider bar and slide to the very end of the C terminal, and you can see that a 47th residue has been added here. If we click on the abbreviation of cysteine or the number, you can see a moving circle called fireflies zoom in to the location. If you'd like to see it closer, right-click on the cysteine and choose Zoom in on residue, and the entire structure will move closer to allow you a better view. Remember, you can always orient it in the orientation that allows you to see it best. And to return the model to the center, choose the blue button with the 4 arrows pointing in different directions at the top. It's a good idea at this point to save your PDB file again so that you don't lose the work that you've just done. Next, it is important that you run an energy minimization to bring the molecules to their ideal conformation. You can do this by choosing Options, choose Experiment, Energy Minimization. A sim box will appear around your model, and you will see a message that the minimization is running. As it finishes, you will see a message in the section at the bottom showing you it has completed after how many steps and what the final energy is of the model. This process will bring the structure of the protein to its most stable configuration.